The light goes out at 200 feet, not gradually, the way dusk falls across a field or fades at the edge of an open door. It goes out in the way a sound stops, present, and then simply not. The ocean above closes over it. The water column above holds 300 feet of pale diminishing blue, and then the blue is gone, and what remains is a darkness so complete that it has no edges, no near side, no far side, only pressure and cold and the slow mineral weight of the Pacific bearing down from above. The submersible descends through this. It is called Nautil, and it is a small white vehicle, blunt-nosed and deliberate, carrying 3 people inside a sphere of steel less than 7 feet across. Outside the hull, the water temperature has already dropped below 40 degrees. The pressure against the hull is equivalent to a column of water more than a mile tall, pressing inward from every direction at once. None of this is felt inside. The 3 people sit in a space barely large enough for all of them, watching their instruments, watching the darkness through small, thick windows, descending at roughly 100 feet per minute through water that has not seen light since before anyone aboard was born. It is March 2005. The vessel above, L'Atalanta, a French research ship operating under the flag of IFRIMER, the French institute that studies and exploits the sea, holds its position on the surface while Nautilon falls. The seafloor here lies roughly 1.5 miles below the ship's keel. Below the ship, below the Nautili, below the water column, the Pacific-Antarctic ridge runs like a slow seam through the crust of the earth, a place where 2 tectonic plates are pulling apart from one another at the pace of a few centimeters each year, and where the interior heat of the planet finds its way upward through cracks in the ocean floor. This region of the seafloor has no name that most people would recognize. It lies approximately 1,500 kilometers south of Easter Island, in a part of the ocean that sees almost no ship traffic, almost no scientific attention, a place whose geography begins and ends in the records of a few research expeditions. The nearest land is an island famous for its stone figures staring out to sea. Below them and below the sea itself, the stone is cracking. The Nautile's exterior lights come on, and a narrow cone of pale white illumination reaches perhaps 30 feet into the surrounding dark before the water absorbs it entirely. The lights do not reveal the seafloor yet. There is only the lit water, particles drifting in the beam like slow snow, the occasional flicker of something bioluminescent retreating from the intrusion of the light, and beyond that, in every direction, the absolute dark of a place that has no day and no season and no cycle of light except the rare and brief one created by human machines descending into it. The instruments register the depth. 1.25 miles, 1.33 miles. The pressure continues to build in the water outside the hull, and the hull continues to bear it, and the 3 people inside continue to descend toward a seafloor that most of their colleagues have never seen, and that no submersible has ever visited at these precise coordinates. At roughly 7,200 feet of depth, the seafloor appears. It comes into the edge of the lights first, as a faint pale suggestion, a slightly different quality of dark below the dark, and then as texture, then as shape, and then suddenly as a specific place, a field of dark basalt rubble, broken and angular, the remnants of lava flows that cooled against seawater in past episodes of volcanic activity along the ridge. The rock is dark gray and dark brown and nearly black. It is covered in places by thin dustings of pale sediment that drift and shift faintly in the thermal convection rising from below. The seafloor here is not flat. It is broken and uneven, heaved and faulted, tilted in places, cracked through in others, and from some of those cracks, A faint shimmer rises, the heat distortion of water several degrees warmer than the ambient seawater around it, water that has spent time somewhere near the interior of the Earth. This is the Easter Island microplate vent field. It is an area of active hydrothermal venting along the Pacific Antarctic Ridge, and it is, in the vocabulary of deep-sea biology, an oasis. In the surrounding seafloor, the abyssal plain, where almost no sunlight-derived organic material reaches and where organisms must survive on what drifts slowly down from above, life is sparse and slow and cold. But here, where the Earth's interior heat drives chemical reactions in the water, life is dense and warm and active in a different way entirely. Not in the way of a sunlit reef, with its visible colors and its constant darting motion, in a slower way, a chemical way, a way that has been operating in the dark for longer than most of the ocean floor has existed. Nautile moves carefully above the basalt field, its thrusters making small corrections, its cameras recording, and in the cone of its exterior lights, against the dark rubble of a seafloor no one has looked at before, there is something pale. It is pale in the way that things become pale when they have lived for a very long time without light, not the pale of something sickly, but the pale of something that has simply had no use for pigment, no reason to be any color at all. The body is a whitish cream, faintly yellowish, and it is resting on the dark basalt with the absolute calm of an animal that has no predators here, that it has not already learned to manage. It is roughly 6 inches long, measured from the front of its carapace to the rear. Its walking legs are tucked partially beneath it, and its claws, its calipeds, the paired appendages that reach forward from the front of its body, are extended slightly outward, and they are covered in something that the camera, even at this distance, registers as soft, dense, silky, a coat of fine yellowish-white bristles that covers the calipeds along their entire length, giving them a furry appearance entirely unlike the smooth, hard-surfaced claws of any crab familiar to anyone watching the footage above. On the research ship, where the scientists are leaning toward the monitors and going quiet, the animal does not move. It holds its position on the dark stone with the patience of something that has no awareness of being seen. Its claws remain extended. Its body remains still. The thermal shimmer of the vent field rises around it and distorts the camera image faintly at the edges, and the pale form at the center of the frame remains exactly where it is, soft and strange and entirely itself, in a place where no one expected to find anything like this at all. There are good reasons why no one expected this. At these depths, in these pressures, in this perpetual cold dark, the biology of the deep sea follows patterns that have taken decades of research to begin to understand. Crustaceans live here, shrimp, crabs, amphipods, isopods, but they belong to known families, known genera, known species, or close relatives of known species. The deep sea is not a place where entirely new branches of the crustacean tree of life are likely to appear. The ocean has been studied long enough, and the crustaceans are numerous and well catalogued enough, that discovering an animal so different from all known forms that it cannot be placed within any existing family is an event that happens, if it happens at all, perhaps once or twice in a generation of scientists. Not often, not casually. It requires not just an unfamiliar face, but an unfamiliar body plan, proportions, structures, and anatomical details that do not match any box that has already been made. The pale furry animal on the basalt rubble did not match any box. It was collected carefully. With the manipulator arms of the nautile, it was lifted from the stone where it had been resting and placed in the collection container mounted to the exterior of the submersible, and it began its long, slow ascent back toward a world of light and air and pressure that it had never experienced. By the time the noddle reached the surface and was recovered by Latalanta, the animal was in preserving fluid, its chelipeds pressed gently against the glass of the specimen container, the dense yellowish bristles still clearly visible, the pale body still holding something of the shape it had maintained on the seafloor. Aboard the ship, under bright laboratory lighting entirely unlike anything it had ever encountered, the morphological documentation began. The scientists who examined it were looking for a place to put it. That is, in a practical sense, what taxonomists do. When a new organism is collected, the first task is placement, figuring out where in the existing map of life this thing belongs. The map is organized hierarchically, from the broad and ancient to the narrow and recent, kingdom, phylum, class, order, family, genus, species. For a crustacean from the deep Pacific, the order is almost certainly Decapoda, the 10-footed crustaceans that include crabs, shrimp, lobsters, and their relatives, a group containing many thousands of known species. Within Decapoda, the infraorder Anemura, sometimes called the false crabs, a group that includes hermit crabs, squat lobsters, and their relatives, seemed from external appearance to be a reasonable starting point. The animal had the right general proportions, the right arrangement of legs and appendages, the right relationship between body and tail. But beyond that rough placement, something stopped working. The setae, the bristles covering the chelipeds, were wrong in the sense of being unlike anything in the existing literature. Not merely long, not merely dense, but architecturally distinct from any sensory or grasping function that setae normally perform on crustacean appendages. The distribution across the chelipeds was unusual. The structure of the chelipeds themselves departed from known anomuran patterns in ways that accumulated into something significant. And the more carefully the specimen was examined, the more clearly it became that placement within any existing anomuran family would require ignoring too many details that did not fit. A family is not a trivial taxonomic unit. At the species level, new discoveries are common. Hundreds of new species are described each year across all groups of organisms. At the genus level, novelty is less common, but not rare. At the family level, a genuinely new family requires evidence that the organism represents an independent evolutionary lineage, old enough and distinct enough that no existing family can accommodate it, that the branch it sits on diverged from all known branches early enough and traveled far enough since that the gap between it and its nearest relatives is as wide as the gap between, say, a true crab and a hermit crab. That is a large gap. Establishing that it exists for a new specimen requires careful morphological work and the willingness to accept that the world contains something genuinely new rather than something merely unusual. The scientists accepted this, and so the formal work of description began. Marine biologists Michel Segunzac, Matthew McPherson, and Bertrand Afonso took the specimen, their field notes, their morphological measurements, and their photographs and observations, and assembled them into a formal scientific description, the document that establishes, for the permanent record of biological knowledge, exactly what a new species is, what distinguishes it from everything already known, what it should be called, and where it belongs in the hierarchy of life. This kind of document follows conventions that have been in place for more than 200 years, going back to the Linnaean tradition of systematic nomenclature, and its publication in a scientific journal represents the moment a new species officially enters the map of life. Before publication, the animal is a specimen. After, it is a species. The paper was published in 2006 In the journal Zoosystema, it established a new genus, Kiwa, named for the Polynesian goddess of shellfish, a name chosen in recognition of the Polynesian geography of the discovery site, the vast Pacific world above the ridge where the animal was found. The species epithet, hirsuta, comes from the Latin for hairy, in direct reference to the dense setae covering the chelipeds that had made the animal so distinctive in the camera footage and so resistant to placement in any existing family. Kiwa hirsuta, the hairy kiwa, and around the new genus, a new family was formally established, Kiwidae. The word Kiwidae has a specific formal meaning in the structure of biological taxonomy. The suffix idae marks it as a family name, following the Linnaean convention applied to all animal families. Its appearance in print in Zoosystema in 2006 marked the creation of a new branch on the crustacean tree of life. Not a twig, not a new leaf on an existing branch, but a branch itself, a lineage distinct enough from all other anomaran lineages that it required its own name and its own place on the map. New crustacean families are not described often. The history of deep-sea exploration has produced them occasionally when submersible technology allowed access to habitats previously impossible to reach. When molecular techniques revealed that animals long considered close relatives were actually deeply separated, when collections from poorly explored regions turned up organisms that did not fit. But they remain rare events, each one a significant revision to a map that had seemed, from a certain angle, nearly complete. The pale animal found on the basalt rubble of the Easter Island microplate vent field, resting calmly in the cone of the Nautil's exterior lights at 7,200 feet, with its dense yellowish bristles and its cream-colored body, and its extended furry chelipeds, turned out to be not merely a new species, but the founding member of a new family, a lineage with no living relatives yet documented, occupying a position on the crustacean tree that had been empty until that March of 2005, simply because no one had looked in quite this place before. It is worth holding that thought still for a moment, the way you hold a small found object to look at it before putting it down. The ocean is vast. It covers most of the surface of this planet, and the largest part of it lies below the reach of light, in depths that have almost never been visited, and that contain, almost certainly, many things not yet documented, not yet named, not yet placed anywhere on any map. The tools for visiting those places have existed for less than a century. The submersibles capable of reaching the deepest known hydrothermal vent fields were built within living memory. The institution of taxonomy itself, the formal naming and placing of organisms, has been operating in its current form for roughly 2.5 centuries, and it is working against a biological diversity that is, by any reasonable estimate, still largely uncounted. Most species on Earth have not been found. Most species in the deep ocean have probably not been found. The map is being drawn in real time by a relatively small number of people, using tools that are better than they were 50 years ago, but still limited, still dependent on where ships go and how deep submersibles dive, and which seafloor habitats happen to be visited on which expeditions. The crab found on the Easter Island microplate vent field in March 2005 was found because L'Atalante happened to be operating in that region, because Ifremeker had the resources and the scientific mandate to deploy Nautil to that depth, because the expedition's targets included the thermal vents of the Pacific Antarctic Ridge, and because the lights of the submersible swept across a particular patch of dark basalt rubble at a particular moment during a particular dive. A slightly different path across the seafloor, a different choice of transect, a different day, and the crab would have stayed where it was, untouched and unnamed, known only to itself and whatever else lived in that thermal field, as it had been for whatever length of time its ancestors had lived there, in the dark, farming the invisible chemistry of the vent fluid in ways that no one had yet thought to look for. That is the quality of deep sea discovery that is hardest to communicate without overstating it. It is not that the ocean is full of wonders waiting to astonish, that language is too loud for what it actually is. It is quieter than that. It is that the ocean is simply large and mostly unvisited, and that the map of life, which looks from some angles almost complete, is from other angles barely started. The animals already named and described, the families already established, the phylogenetic trees already drawn, these represent a genuine achievement of centuries of careful science. And they are also, in the context of the actual biological diversity of the planet, a beginning. The seafloor of the Easter Island microplate held something that the entire existing map of crustacean families could not accommodate, and it held it quietly, without announcement, without urgency, the way the deep sea holds everything, in the dark, under pressure, at a temperature barely above freezing, in a world that neither needs nor registers the act of being found. The chelipeds of the specimen pressed against the glass of the preservation jar. The yellowish bristles, still dense, still silky, held their shape in the fluid. Under the bright lights of the laboratory on La Talante, a scientist's hand moved a magnifying lens across the surface of the carapace, tracing the margin of the rostrum, measuring the distance between features, noting the absence of structures that would allow placement within any known anomarin family. The notes accumulated. The measurements formed a pattern. The pattern did not match any existing entry in any existing key to crustacean families, and eventually the person holding the lens put it down, and the understanding settled. This is something new, not merely new in the sense of an unfamiliar face on a familiar body. New in the deeper sense. New family, new. Kiwa. One word in a journal. One new branch on a tree that had been growing for 500 million years. The Pacific Antarctic ridge continues to pull apart, a few centimeters each year, as it has been doing since long before this expedition, and as it will continue to do long after all records of it have been made, and filed, and indexed. The vent field at the Easter Island microplate continues to exhale warm, mineral-laden water into the cold dark of the deep Pacific. And in that darkness, on the basalt rubble of the seafloor around those vents, the animals that were not collected on that March dive in 2005 continued doing exactly what they had been doing before the lights of the nautile swept across them, extending their furry pale chelipeds into the chemical gradient of the rising vent fluid, holding very still, attending to the small and invisible economy of their own bodies in a manner so specialized, so precisely fitted to this one improbable environment that it would take years of subsequent research to understand what exactly they were doing there. The name Kawaii appeared in Zoosystema in 2006, and the map of life was redrawn quietly at the family level. That does not happen often. It happened because a ship went to a part of the ocean it had not visited before, and a submersible descended into a darkness from which it brought back something that nothing in the existing literature had prepared anyone to expect. And the crab, or its close relatives, continued to live where they had always lived, at the margin of the vent field, in the warm dark, at the edge of the Earth's own heat, patient and furry and pale, farming something no one yet had a word for, waiting to be understood. The journal was published. The family name entered the permanent record. Somewhere below the southern Pacific, at a depth where the weight of water is measured in hundreds of atmospheres, A small cream-colored animal extended its claws into a warm current, rising from a crack in the seafloor, and held them there, as still as stone, as old as the ridge, as steady as the slow heat of the interior of the earth, moving its bristled arms in a rhythm so quiet and so small that the instruments of the submersible on that first dive had not caught it at all. The crab rested in the dark. The map held its new name. The ridge continued to open, a few centimeters each year, in a silence too deep for sound. Along the Pacific Antarctic ridge, two plates of ocean crust pull apart from each other at the pace of a fingernail growing. That is all the speed there is. A few centimeters every year, the rock separating along a seam that runs for thousands of miles beneath water no sunlight has ever reached. The pulling is silent. The rock does not announce it. Deep in the crust, where the planet's interior heat presses upward, the separation opens channels too thin to see, and cold seawater, heavy with pressure, begins to move down through them. The water descends. It moves through cracks in the basalt, through hairline fractures and widening fissures, down toward the heat. The deeper it goes, the hotter the rock becomes. The water does not boil. The pressure above it is too great for boiling in the ordinary sense, but it grows hotter with every meter it descends, and as it heats, it begins to change. The minerals in the surrounding rock dissolve into it. Iron passes into solution. Manganese, copper, zinc, sulfur compounds shift and reform. The water that entered the crust as cold, relatively clean seawater is being transformed, slowly and continuously, into something chemically far stranger, a fluid that carries the dissolved interior of the earth itself. This process has been running since the ridge first opened, which is to say, it has been running for millions of years without stopping once. The superheated fluid, now laced with hydrogen sulfide, heavy with dissolved metals, stripped of the chemistry it arrived with, eventually finds its way back to the surface of the ocean floor. It rises through the crust on the heat that drives it, following the same pressure gradients that drew the cold water down. Where the crust is thin and the channels are large, it emerges with force, shooting upward into the near-freezing ambient water at temperatures that can exceed 400 degrees Celsius. The moment it contacts the cold ocean, the dissolved metal sulfides precipitate out of solution almost instantly, forming dark mineral clouds that billow upward from the orifice in a continuous dark plume. These are the black smoker vents, tall mineral chimneys built by the same precipitation, layer by thin layer, over decades and centuries, rising several meters from the seafloor in dark columnar structures that exhale their mineral plumes without ceasing. The chimney walls are their own record. Each thin mineral layer is a day's worth of precipitation. The tallest chimneys are the oldest, and some have been building themselves since before any human being thought to look for them. Where the crust is thicker and the channels are narrower, the same fluid emerges more slowly, seeping upward through diffuse pathways at lower temperatures, sometimes only a few degrees above the ambient water, sometimes tens of degrees warmer. These diffuse flow zones spread across broader areas of the seafloor, and their temperatures are variable and patchwork, creating a landscape of invisible thermal gradients that shift from place to place across a vent field, the way warmth and coolness shift unevenly across a room where a single window lets in winter air. The chemistry shifts accordingly. In one patch of seafloor, hydrogen sulfide seeps upward in high concentration. A meter away, the concentration drops. Dissolved oxygen, arriving from the ambient water above, diffuses downward and meets the rising sulfide somewhere in the middle, at some depth that depends on how much of each is present at that moment in that place. This meeting of chemical opposites, sulfide rising, oxygen descending, is where the life begins, on the rocks and sediments of the vent field, in the warm water just above the diffuse flow zones and along the flanks of the tall black smokers. There are films of bacteria. They are visible in places as pale, whitish mats that cover the rock surface, the way a thin layer of frost covers pavement, irregular and patchy, following the contours of the diffuse flow beneath them. In other places, they are too thin to see without magnification, a single layer of cells pressed against the mineral surface, each cell processing the chemical transaction that sustains it. The transaction is simple in outline, though the molecular machinery that performs it is ancient and intricate. Hydrogen sulfide arrives. Oxygen or some other electron acceptor is available. The bacterium captures the energy released when one reacts with the other and uses that energy to pull carbon dioxide from the surrounding water and assemble it into organic carbon, into the molecular architecture of life. The bacterium makes food from chemistry. It does not need light. It does not need any connection to the surface of the ocean or the atmosphere above it. The sun is 2,000 meters above and might as well be in another universe entirely. What the bacterium needs is a gradient, a difference between the chemical richness of the vent fluid and the relative poverty of the ambient water, and the gradient is always there as long as the vent is active, which is to say, as long as the plates keep moving, as long as the heat of the earth keeps pressing upward through the thinning crust. The bacteria fix carbon as long as the gradient holds. The gradient holds as long as the earth produces heat. The earth produces heat for as long as it has a molten interior and a slowly decaying store of radioactive elements, which is to say, for a very long time. The process that turns inorganic chemistry into biological material without any reference to sunlight is called chemosynthesis. It is not a variant of photosynthesis. It is not a simplified version or an early evolutionary precursor or a degraded form. It is a complete and independent solution to the problem of building organic matter from available raw materials, arrived at in the dark, powered by the same electrochemical logic that powers all metabolism everywhere, but drawing on a different energy source entirely. Where photosynthesis captures energy from photons, chemosynthesis captures energy from chemical reactions, the movement of electrons from sulfide, or methane, or hydrogen gas to an electron acceptor, a transaction that releases just enough energy to turn carbon dioxide into sugar and sugar into everything else. The vent field around the Easter Island microplate holds this process running continuously across its extent, across every bacterial mat, every thin film of cells on the chimney walls, every microbial community tucked into the porous rock surfaces at the vent margins. The scale of it is invisible because most of it is invisible, but the consequence of it is everywhere. Life at the vent is thick. That is the first thing the submersible cameras showed when researchers first began exploring active vent systems along the East Pacific Rise decades ago, and it was not what anyone expected to find. The surrounding deep sea floor, away from the vents, is nearly empty, a flat, cold expanse of soft sediment where organic material drifts down from the surface ocean at a trickle and is consumed almost as fast as it arrives. The biomass density there is low and spread thin. Then the submersible reaches the vent field, and the picture changes entirely. The rock is covered. The water above it is dense with movement. Animals cluster around the vent emissions in numbers that recall shallow coastal ecosystems rather than the arid deep sea. What feeds all of this is the bacteria and the chemosynthesis they perform. At the outer margins of a vent field where the thermal influence is weakest and the chemistry most diluted, pale bacterial mats spread across the sediment in uneven patches. The water here is only slightly warmer than ambient, and the hydrogen sulfide concentration is low. But even at low concentrations, the bacteria find enough gradient to sustain themselves. In the spaces between the mat patches, smaller organisms graze, tiny crustaceans, polychaete worms, nematodes, feeding on the bacteria directly or on the organic material the bacteria have produced and left behind in the sediment. Moving inward toward the stronger emission zones, the temperature and chemistry become more intense and more variable. Here the larger animals concentrate. The tube worms called Riftia pachyptilla stand in clusters above active diffuse flow areas, their tubes anchored to the rock and their bodies extended upward into the water column, each individual topped with a bright red plume of gill filaments. The red comes from hemoglobin, a special form of it that binds both oxygen and hydrogen sulfide simultaneously and carries both to the bacteria living inside the tube worm's body in a dense tissue called the trophosome. The tube worms have no mouth, they have no gut. In the adult form, they do not eat anything in the external sense of the word. Everything they need comes from the bacteria within them, which chemosynthetically produce enough organic carbon to sustain the entire worm. Each individual is a collaboration, an animal body built as a delivery system for bacteria that have never needed to be outside. The tallest of these tube worms reach 2 meters in length. They are the largest known animals that rely entirely on internal chemosynthetic bacteria for their nutrition, and they grow at rates that seem impossible for the deep sea, several centimeters in length per year, in an environment where most organisms grow slowly and most food is scarce. The food at the vent is not scarce, the bacteria see to that. Clustered near the tube worm beds, and around the bases of black smoker chimneys, and in the warm diffuse flow areas at vent margins, are mussels with their own chemosynthetic cargo. The genus Bathymedoilus, a group of deep-sea mussels related to the mussels of shallow coastal waters, houses chemosynthetic bacteria in the cells of its gill tissue. Unlike the tube worms, the mussels retain some digestive capacity and can supplement their bacterially produced nutrition by filter feeding on particles in the water. But their primary food source is the same, organic carbon produced by bacteria inside their own bodies from the chemical energy of the vent. These partnerships between animals and chemosynthetic bacteria, where bacteria live inside animal tissues and the animal provides them with shelter and raw materials, while the bacteria provide organic carbon in return, are among the most tightly integrated biological relationships known. The animal cannot survive without the bacteria. In many cases, the bacteria cannot survive outside the animal tissue. They have been living together long enough that neither is quite what it would be alone. The thermal landscape they all inhabit shifts across the vent field in ways that matter deeply to every organism present. Right at the orifice of a black smoker, where vent fluid emerges at hundreds of degrees, nothing that could be called macroscopic life persists. The temperatures there are close to the range, tolerated only by thermophilic microorganisms, archaea that have evolved to function at temperatures that would denature the proteins of ordinary cells in seconds. Methanopyrus candleri, an archaean found in superheated vent zones, can grow at temperatures approaching the upper limit of known life. These organisms belong to a part of the tree of life that is entirely their own, ancient lineages that diverged from the rest of life very early, adapted to extremes that the rest of life has never occupied. A meter away from the orifice, the temperature might be 50 degrees lower. 2 meters away, lower still. The mixing of vent fluid and ambient water is turbulent and uneven, and the thermal contours around a single vent shift minute by minute as the flow rate varies and as currents in the ambient water change direction. An organism living in the warm mixing zone exists in a chemical and thermal environment that is never quite the same twice. The hydrogen sulfide concentration at one point on the rock might change by a factor of 10 within a few minutes as a pulse of vent fluid passes and then thins into the ambient water. The organisms that have persisted in this landscape have done so because they can tolerate this variability, or because they have found some way to use it. Along the flanks of vent structures and in the moderate temperature zones where diffuse flow emerges, a different class of small crustaceans has been present at vent sites since the first systematic surveys. Brazilian shrimp swarm in some Atlantic vent fields in densities that darken the rock surface, their pale bodies pressed together in slow-moving masses. Some of these shrimp carry chemosynthetic epibond bacteria on the surfaces of their bodies, not inside their tissues, as in the tube worms and mussels, but on the outside, attached to external surfaces and sustained by the vent chemistry that diffuses through the water the shrimp inhabits. The shrimp, in turn, may scrape and consume some of this bacterial growth. The biological logic of the vent, played out across many different lineages, returns again and again to the same arrangement: an animal positioned in the chemical gradient, bacteria processing the gradient on or within the animal, organic carbon moving from bacteria to animal. The details differ. The evolutionary pathways that led each lineage to this arrangement are entirely separate. None of them arrived here by any plan. Each arrived because, over a long time, the individuals that happened to survive in this environment were the ones whose biology was compatible with it, and the ones whose biology was most efficient at using what the vent provided left more descendants than those whose biology was less efficient. Over generations, this process sorted and refined until the organisms living at these vents today are the ones whose bodies are most precisely fitted to the conditions that have always been here. The vent field itself is not stable on the timescale of millions of years. Black smoker systems can be active for decades and then go quiet as the fluid pathways in the crust shift. When a vent system shuts down, the organisms that depended on it must either move to an active site or die. The evidence suggests that vent species do move, that their larvae drift in the deep-sea water column, carried by currents across the distances between active vent fields, settling when they detect the chemical signature of a new active site. The chemical signatures are distinctive: hydrogen sulfide in concentrations far above background, warmth, and the particular dissolved chemistry of vent fluid. Larvae capable of detecting these cues and following them toward their source would have a tremendous advantage over those that could not, and there is evidence that many vent larvae possess exactly this chemosensory ability. The Pacific Antarctic Ridge is a slow-spreading ridge, which means the plates on either side of it are separating at a relatively modest rate compared to some other ridge systems. Along faster-spreading ridges, such as the East Pacific Rise further north, the increased magmatic activity sustains more vigorous and more densely distributed vent systems. Along the slower-spreading Pacific Antarctic Ridge, vent fields are more widely spaced, and the hydrothermal activity at any given site may be lower in intensity. This matters for the organisms that live there. Lower vent fluid flux means lower concentrations of hydrogen sulfide in the water column above the diffuse flow zones. Lower concentrations of hydrogen sulfide mean a less chemically rich environment for chemosynthetic bacteria, and a less chemically rich bacterial community means lower primary productivity, less food for the animals that depend on it, and yet life is present. The vent field at the Easter Island microplate, sitting roughly 1,500 miles south of Easter Island on the Pacific Antarctic Ridge, supports a community of organisms. The submersible that descended to this field in 2005 found it occupied. It found the bacterial mats on the rock surfaces. It found vent fauna present and active. It found something new. What it found had pale cream-colored limbs and a body roughly 6 inches long. It moved slowly across the dark basalt rubble near a diffuse flow zone. Its chelipeds, the front limbs that in most crabs bear claws, were extended slightly from its body, and they were covered in dense, silky yellowish-white filaments that caught the submersible's lights and held them for a moment before the animal moved again. No one looking at the footage from that submersible had seen anything quite like it. The discovery itself belongs to an earlier part of this story. What matters here is the environment into which this animal had arrived, or rather, the environment in which it had always been, long before there were any submersibles to find it. The vent field had been running its chemistry continuously, building and dissolving and rebuilding its community of organisms for as long as the ridge had been active at this location. The animal's ancestors had been present for some portion of that time, finding their way to this thermal and chemical landscape, and gradually, over generations, becoming more precisely fitted to it. The vent did not change for the animal. The animal changed around the vent. Everything present in the vent field exists in a relationship with the chemistry that the Earth produces there. The chemical gradient between the sulfide-rich vent fluid and the oxygen-bearing ambient water is the axis around which the entire community organizes itself. Bacteria occupy the gradient directly, metabolizing it, living at the precise interface where one chemistry meets the other. Animals occupy positions relative to the gradient, closer for those that can tolerate higher sulfide and temperature, farther for those that cannot, and at the specific position that maximizes their access to whatever food source they have evolved to exploit. This organization is not planned or coordinated. It emerges because every organism present is drawn toward the conditions under which it functions and repelled from conditions that damage it. The result looks almost designed, a series of concentric zones, each inhabited by organisms with the thermal and chemical tolerance appropriate to that zone, but it arises from the independent movement of each individual following its own chemistry. The tube worms are where the tube worms are because their larval ancestors settled where the conditions suited them and grew and reproduced. The mussels are where the mussels are for the same reason. The pattern is real. The plan is not. On the outer edge of the diffuse flow zone, where the gradient is gentlest and the temperature barely above ambient, the bacterial mats thin out and the water becomes progressively less chemically rich. Here the vent's influence fades into the surrounding deep sea. The organisms present at this margin are those capable of living at the edge of the gradient, capturing just enough of the vent's chemical energy to sustain themselves, while also tolerating the cold and relative chemical poverty of the ambient deep-sea water that constitutes most of their environment. This is approximately where Kiwa hirsuta is found. Not at the hottest part of the vent, where the chemistry is intense but the temperatures approach the limit of what crustacean physiology can tolerate. Not at the very outer margin, where the vent's influence is too dilute to support the bacterial growth that the animal depends on. It occupies the warm but not lethal zone, the chemically active but not overwhelming zone, the region of the vent field where the gradient between sulfide and oxygen is steep and variable and productive, where bacterial growth is fastest and most sustained. To be in this zone is to be in a place that is simultaneously warm and cold, sulfidic and oxygenated, depending on which current is passing at any given moment. The vent fluid rises and then dilutes. The ambient water sweeps in from the sides. A position that was warm a minute ago may be cool now. A position that was sulfide rich may be briefly oxygenated and then sulfide rich again. To metabolize efficiently, the bacteria living at this interface need to experience both: enough sulfide to fuel their chemosynthesis, enough oxygen to serve as the electron acceptor the reaction requires. Neither resource alone is sufficient. The gradient between them is what makes life possible for these particular organisms. The whole of the vent ecosystem is, in this sense, a system organized around a gradient. And gradients require difference: one high concentration meeting one low concentration, one warm zone meeting one cool zone, one chemical richness meeting one chemical poverty. Without the difference, there is no gradient. Without the gradient, there is no driving force for the chemistry. Without the chemistry, there is no food. And without the food, there is no life in numbers that would be visible from a submersible. The Earth maintains this difference continuously. Magma presses upward beneath the ridge. Cold seawater presses downward into the crust. The two systems never equilibrate because the heat from the planet's interior is constantly renewed by the slow decay of radioactive elements in the rock and by the residual heat of the planet's formation. 4.5 billion years ago, a warmth that has been leaking into the ocean floor in pulses of hydrothermal activity ever since. The vent field at the Easter Island microplate is one point in a global system of mid-ocean ridges that circles the planet like the seam of a large irregular sphere. The total length of the mid-ocean ridge system is enormous, roughly 45,000 miles, running through every ocean basin. Along this entire seam, in varying degrees, the same basic geological process operates. Plates separate, magma fills the gap, seawater circulates through the crust, and emerges transformed. The hydrothermal chemistry happens everywhere along this seam, and wherever it happens, the gradient exists, and wherever the gradient exists, chemosynthetic bacteria are present to use it. Some of these bacterial communities exist in isolation from one another, in vent fields separated by hundreds of miles of cold, nearly lifeless deep-sea floor. The organisms at one vent field may have had no biological contact with organisms at a distant vent field for thousands of years. Their communities may have developed independently, with different species present, different community structures, different assemblages of organisms adapted to the specific chemistry and temperature conditions at that particular site. And yet the basic logic of the ecosystem, bacteria metabolizing the chemical gradient, animals consuming the bacteria or housing them as internal partners, repeats at vent after vent in every ocean wherever the conditions are right. The same solution found again and again in different places by different evolutionary paths. Chemosynthesis as a metabolic strategy is not confined to hydrothermal vents. It operates in other environments where chemical gradients exist. In the sediments of cold methane seeps on the continental shelf, where methane oxidizing bacteria sustain their own communities of mussel and tubeworm relatives. It operates in sulfide-rich coastal sediments. It operates at low rates in many places where anoxic and oxic zones meet. But the hydrothermal vents concentrate it. They produce the steepest, most sustained, most productive chemical gradients on the deep sea floor, and they have been doing so for at least several 100 million years. Whatever the earliest history of chemosynthesis, and there are serious proposals that it may represent one of the oldest metabolic strategies in the history of life. The vents have been one of its great theaters. The black smoker chimneys are built by the very chemistry they exhale. Each pulse of superheated fluid carries dissolved metal sulfides into the near-freezing water at the chimney mouth. The sulfides precipitate. The precipitation deposits mineral on the chimney wall. The wall grows. Over years and decades, the chimney extends itself upward, built from the material that the fluid inside it is shedding as it cools. The chimney is not built toward anything. It extends upward because the mineral precipitation follows the temperature gradient, and the temperature gradient at the chimney mouth is always at the top where the hot fluid first meets the cold water. What appears to be purposeful construction is purely the behavior of chemistry in a thermal gradient. The same logic, the same absence of direction, applies to everything in the vent ecosystem. The bacteria grow where the gradient feeds them. The mussels settle where the water chemistry suits their gill bacteria. The tubeworm larvae descend through the water column and settle on rock surfaces that their chemoreceptors identify as suitable, and the worms that grow from those larvae are anchored for the rest of their lives. Nothing in the system is choosing in any meaningful sense. Every organism is responding to the conditions immediately around it, and the sum of all those responses is a community of extraordinary complexity and density, organized around a heat source in the dark floor of the Pacific Ocean. This community had been present, in some form, at the Easter Island microplate vent field long before the submersible arrived. The organisms living there were not waiting to be discovered. They were occupied entirely with the business of the gradient, metabolizing it, reproducing, competing for position in the warm zones, maintaining their epibiont bacteria, processing sulfide through their gill tissues, brooding their eggs in the cooler water at the vent margins. The vent field was complete and sufficient in itself, running on the Earth's own chemistry, without any connection to the world of sunlight and photosynthesis 2,000 meters above. When the submersible's lights first fell on that part of the sea floor, nothing changed in the community. The lights were strange and temporary. The submersible was a brief visitor. The animals may have responded to the light and the disturbance of the vehicle's movements in the water, but the vent kept flowing. The bacteria kept growing on every available surface. The chemical gradient kept rising from the diffuse flow zones and meeting the descending oxygen. The community continued. The pale-armed animal at the edge of the diffuse flow zone, waving its dense cetoschelopeds slowly through the water, was part of this community in the same way everything else was, positioned precisely in the gradient that sustained its particular biology, doing what it had always done in the dark. It did not know there was a surface above the ocean or a sun above the surface. It knew only the chemistry of the water around its outstretched arms, the warmth rising from below, the faint mechanical vibration of the current, and the gradient was there. The sulfide rising, the oxygen descending, the bacteria growing, slowly and continuously in the space between. That space, the thin, variable, chemically rich zone where vent fluid meets ambient seawater, is the foundation of everything the vent supports. It is the original energy source, the first step in the only food chain present, the reason the tubeworms grow tall, and the mussels cluster thick, and the small crustaceans congregate in their hundreds around the warmest diffuse flow zones. The sun powers none of it. The rain feeds none of it. The carbon fixed in the leaves of trees and the plankton of the surface ocean does not reach this depth in any quantity that matters to these organisms. They are sustained entirely by what the earth makes for itself, by the chemistry of its own interior, leaking upward through the crust in steady, mineral-rich, sulfide-bearing flows, meeting the cold and the oxygen of the deep ocean in a transaction that has been producing organic matter in the dark for longer than there have been eyes to see it. The plates keep moving, the heat keeps rising, the gradient holds. The bacteria grow on every surface the warm water touches, in every crack and fold of the basalt, on the chimney walls and the sediment and the rock rubble of the vent field floor. They grow on whatever is present in the mixing zone, on the mineral surfaces, on each other, on the surfaces of the animals that have learned to position themselves in the gradient's path. In the slow, still dark at the bottom of the Pacific, the chemistry of the earth converts itself continuously into life. The vent field does not require a witness. It does not require acknowledgement or record. It produces its bacterial mat and its tube-worm community and its pale crustaceans and its mineral chimneys with the same patient consistency it has always brought to these things, drawing on a source of energy that has nothing to do with the star at the center of the solar system, operating on the slow internal logic of a planet that has been generating its own heat since before there was any life on its surface, to take advantage of it. The process continues in the dark. It will continue after the lights have gone. The gradient rises, the bacteria grow. The water above the vent field is faintly warmer than the water beside it, and in that faint warmth, in that slight chemical difference between one column of water and the next, the entire ecosystem holds itself quietly together, as it has always done, as it will continue to do, long and slow and deep and still. Under an electron microscope, a single seta looks like a country road, seen from far above, long, narrow, continuous, stretching from one end of the frame to the other with no visible terminus in either direction. But the surface of that road is not bare. From the base of the filament to its farthest tip, pressed close along every side, bacterial cells sit in dense and unbroken community, each one smaller than a wavelength of visible light, each one living its small chemical life against the seta's wall. The filament itself is no wider than a few thousandths of a millimeter, and yet it holds hundreds of cells, perhaps thousands, packed so tightly that the seta's own surface is almost invisible beneath them, buried under a continuous mat of living tissue that the microscope renders as a kind of soft gray fur on an already furry limb. This is a single seta. Kiwa hirsuta has thousands. They cover the chelipeds, the large forward-reaching claws, in dense overlapping rows, each seta a column of bacteria unto itself, each column running parallel to its neighbor in the close-packed array, unto itself, each column running parallel to its neighbor in the close-packed array. The whole surface of the claw transformed into something between a brush and a garden, a biological structure whose architecture does not match anything known from sensory bristles or grasping hairs in other crabs. Sensory setae are sparse, widely spaced, designed to catch a signal from the water and carry it inward to a nerve. Grasping setae are rigid, curved, built for mechanical work. These are neither. These are long and fine and densely, evenly distributed, and under the electron microscope, their surfaces bloom with bacteria from base to tip, every millimeter of every filament colonized and covered and alive. To see a single one of these setae is to see almost nothing of the animal. Pull back from the microscope's image, and the setae becomes part of a claw, and the claw becomes part of a body 15 centimeters long, pale as cream against the dark seafloor. Its two large chelipeds held slightly forward and slightly raised, the yellowish-white fur of bacteria-covered setae catching the faint current that rises from below. The crab does not hold still. It moves its claws in slow arcs through the water above the vent, back and forth, in a motion that at first appears almost restless, almost idle, as though the animal were simply treading in place, letting its limbs drift on the current. But the motion is not idle. The motion is the whole point. The water above an active vent is not uniform. It moves in layers, not clean, defined layers with sharp edges, but gradients, shifting bands of chemistry and temperature that change over distances of centimeters and fractions of centimeters. Close to the vent's emission, where warm fluid rises through the basalt, the water carries hydrogen sulfide, methane, and other dissolved compounds that the Earth's interior has produced and the vent is releasing. Farther from that zone, where the ambient water of the deep ocean presses in from all sides at a temperature just above freezing, dissolved oxygen arrives from above, carried downward from the surface by the cold, dense, slowly sinking water that has spent time in contact with the atmosphere before it descended to this depth over months and years. These two chemical worlds, the sulfitic water rising from the vent and the oxygen-bearing water pressing in from the surrounding abyss, do not exist in separate chambers. They meet. They mingle along a gradient that shifts with every small change in vent emission rate, every slight movement of current, every thermal pulse from below. And somewhere in the middle of that gradient, not too close to the sulfide source, not too far into the cold oxygenated water, is a zone where both chemicals are present in concentrations sufficient for a particular kind of metabolism to proceed. The bacteria on Kiwa hirsuta's setae are chemosynthetic. They belong primarily to a group called the epsilon proteobacteria, a lineage of microorganisms that appears repeatedly at hydrothermal vents and other sulfitic environments around the world. Their cellular machinery built around the oxidation of hydrogen sulfide. To do this work, they need two things. They need the sulfide itself, rising from below, and they need an electron acceptor, oxygen or nitrate, to receive the electrons that sulfide gives up when it is oxidized. Without both ingredients, the metabolism slows and eventually stops. The bacteria on the crab's setae are in this chemical sense entirely dependent on being positioned in the mixing zone where sulfide and oxygen overlap. The crab provides that positioning. It moves its claws through the water column in slow, rhythmic sweeps, carrying the bacteria first into sulfide-rich water and then back into oxygenated water, first one way and then the other, the whole bacterial community on each setae cycling through the two chemical environments in steady alternation. On the inward stroke, the setae move toward the vent into the warmer, chemically richer fluid. On the outward stroke, they return to the cooler, oxygen-bearing ambient water. The bacteria experience this as a regular, dependable alternation of the two things they need. They are, in this way, being fed. This is what the wave motion is. Not restlessness, not drift, a method. The animals nearest the vent do not perform this waving motion with the same amplitude as those positioned farther from the emission. The ones closest to the vent tend to hold their chelipeds more extended, moving them through smaller arcs, as though the local chemistry already provides a sufficient gradient without needing to sweep widely. The ones positioned at the outer margin of the vent's thermal influence sweep more broadly, reaching their claws farther into the ambient water column to find the oxygen that does not arrive naturally at their position. Each crab, positioned where it is, seems to calibrate the arc of its waving to the chemistry immediately surrounding it. Though without any awareness of chemistry or calibration, only behavior refined over a long lineage until it matched the physics of its environment with quiet precision. The bacteria do not know they are being moved. Motion is not a category available to a cell that lacks a nervous system, but the consequence of the motion is growth and reproduction and the slow accumulation of more bacterial cells along the length of each seta, the biofilm thickening gradually as the colony expands and the conditions remain favorable. Time passes in the darkness. The crab waves its claws. The bacteria divide. The seta become more densely covered, more productive, more rich with the carbon that chemosynthesis fixes from the surrounding water, organic carbon built from the inorganic, from carbon dioxide transformed by chemical energy into biological tissue, into food, because the crab eats them. This is the part the organism's appearance does not immediately suggest. It is an animal with large furry claws and a body pale as unbaked dough, positioned at a hydrothermal vent 2,000 meters below the surface of the Pacific Ocean. It could be a filter feeder, drawing particles from the water with fine appendages. It could be a scavenger, processing the bodies of other animals that drift or fall. It could be, in any number of ways, simply a resident of the vent community, eating whatever the vent community provides. But when scientists examined its mouth parts under magnification, what they found were structures not built for passive reception or opportunistic scavenging. They were built for harvest. Along the ventral surface of the crab, positioned near the mouth, and arranged to work in coordination with the larger chelipeds, are smaller appendages bearing fine comb-like rows of dense bristles, structures suited to drawing across the surface of the cetos claws and removing the accumulated biofilm that has grown there. Picture a comb drawn through hair, except the hair is bacterial mat and the comb is a living appendage moving in a directed, repeating motion. The setae of the chelipeds are long enough and flexible enough that when these oral appendages engage with them, the bristles can strip the surface, bacterial layer with a motion that is closer to harvesting than to combing, removing the outermost, most developed portion of the biofilm while leaving the basal portions of the bacterial community intact, still attached to the seta, still metabolizing, still capable of regrowing the layer that has been removed. It is a sustainable harvest in the most direct sense. Take the top, leave the root, return tomorrow. The bacterial mat regrows. The crab feeds again. This pattern, cultivation, harvest, and regrowth is not common in the animal kingdom. Growing something, tending it, and then eating it requires a particular kind of behavioral loop that most animals never close. Most animals that eat bacteria simply consume them passively, filtering water or ingesting sediment and processing whatever microbial content that sediment contains. What Kiwa hirsuta does is different, and the difference lies in the waving motion that maintains the bacterial community's productivity before the harvest begins. Without the waving, the bacteria would experience a less favorable chemical environment, and the biofilm would grow more slowly. With the waving, the bacteria are regularly supplied with the chemical gradient they need to metabolize, and the biofilm accumulates faster, and the harvest, when it comes, is richer. The behavior before the feeding shapes the quality of the food. This is not something most animals do. The chemical signature of what the crab consumes is legible in its own tissues. When an organism eats chemosynthetically produced carbon, that carbon carries a particular ratio of carbon isotopes, carbon-12 and carbon-13, that differs from the ratio found in carbon fixed by photosynthesis near the ocean's surface. The deep ocean contains a certain background proportion of each isotope, and photosynthetic production has its own characteristic signature, and chemosynthetic production differs from both in ways that are measurable and reproducible. When scientists analyzed tissue samples from Kiwa hirsuta and measured the carbon isotope ratios in those tissues, the numbers they found did not match photosynthetic carbon, did not match surface-derived organic matter, did not match the prey of most deep-sea scavengers. They matched the carbon produced by chemosynthetic bacteria, by the very microbial communities that populate hydrothermal vent ecosystems and colonize the setae of the crab's chelipeds. The animal's own chemistry recorded what it had been eating, written in the language of isotopes, in numbers that did not lie. The nitrogen isotopes, measured alongside the carbon, told a consistent story. Nitrogen-15 accumulates at each step up a food chain. A predator eating herbivores carries more nitrogen-15 than the herbivores do, and the herbivores carry more than the plants they ate. By measuring the nitrogen-15 ratio in Kiwa hirsuta tissue and comparing it to the nitrogen-15 ratio in the bacterial communities on the crab setae, it was possible to estimate how directly the bacteria contributed to the crab's diet. The numbers placed the crab at approximately one trophic step above the bacteria, consistent with an animal that eats bacteria as its primary food source, not as a minor supplement to some other diet. The isotopic data and the morphological evidence pointed in the same direction. The crab was a farmer. The bacteria were the crop. There is a category of behavior observed in a very small number of animal lineages that involves deliberately cultivating other living things as food rather than simply finding and consuming food that already exists. Leafcutter ants cut sections from living plants and carry them underground, not to eat the leaf material itself, but to inoculate it with fungal spores and grow the fungus in cultivated gardens beneath the colony. The fungus they grow is their primary food source. Without the cultivation, without the careful tending of the gardens, without the removal of competing organisms that would otherwise overtake the fungal beds, the colony cannot sustain itself. The farming is not incidental to the ant's survival. It is the ant's survival. Certain termites have developed an almost identical strategy independently, growing a different genus of fungus in underground chambers, harvesting and eating the fungal tissue on a continuous basis. These two groups, the leafcutter ants and the fungus-growing termites, represent separate evolutionary arrivals at the same behavioral solution: grow something first, then eat it. Kiwa hirsuta is a third arrival, not an insect, not a social organism, not one that lives in a colony or builds a chamber or recruits others to the work. A single crab moving its claws through the dark water above a hydrothermal vent, tending a microbial garden that exists on its own body. The scale is entirely different from an ant colony's underground fungal chambers. The substrate is entirely different. The kingdom of life being farmed is different. But the underlying logic, produce and then harvest, rather than simply find and consume, places Kiwa hirsuta in that same narrow category, a category that evolution reaches so rarely that each new member of it is in its way a kind of arrival. The relationship Kiwa hirsuta has with its bacteria is also different in its physical arrangement from the chemosynthetic relationships maintained by other vent animals. Riftia pachyptilla, the large tubeworm that grows in clusters near active vent emissions, carries its chemosynthetic bacteria inside its own tissues in a specialized organ called the trophosome, where the bacteria live and metabolize in permanent residence, never leaving, never exposed to the open water. The tubeworm provides the bacteria with hydrogen sulfide, which it transports in its blood using a specialized hemoglobin molecule, and the bacteria fix carbon and share the product with their host. The bacteria and the worm live as one, their metabolisms intertwined, their relationship defined by permanent physical union. Bathomodiolus mussels, common at vent sites across the world's ocean ridges, carry similar bacterial symbionts inside gill cells, again in permanent intracellular residence, the bacteria embedded in host tissue, and the tissue fed by bacterial productivity. Both the tube worm and the mussel depend on bacteria that they cannot see, that they carry inside themselves like a second metabolism, invisible to the outside observer. Kiwa hirsuta's bacteria are visible. They cover the surface of the animal in a layer thick enough to give the chelipeds their characteristic yellowish color, their pale fur, the quality that made researchers reaching for a name think of a yeti, something large and furred and pale, inhabiting an unlikely place. The bacteria are outside. They are epibiotic, living on the surface rather than inside the host, and their relationship with the crab is consequently different in character from the permanent, metabolically interwoven relationships of the tube worm or the mussel, where the tube worm's bacteria cannot leave and the muscles cannot leave. The bacteria on Kiwa hirsuta's setae are exposed to the surrounding water, potentially subject to being washed away, potentially subject to competition from other microorganisms, potentially dependent on the crab's wave motion behavior to keep the chemical conditions around them suitable for growth. They are a community on a surface, not a partnership sealed inside a body. The crab and the bacteria share a relationship defined by behavior rather than anatomy. This distinction matters for understanding how the farming works. Because the bacteria live outside and are periodically harvested, the crab must actively maintain their community, must continuously perform the waving motion that provides the chemical gradient the bacteria need, and must time and calibrate its harvesting carefully enough that the bacterial population always has the capacity to recover and regrow. An animal that harvested too aggressively, stripping its sedae completely, removing all bacterial cells, would destroy the productivity of its own crop and be left with bare sedae, offering nothing. An animal that never harvested would eventually fill its setae to maximum bacterial density, but would gain no nutritional benefit from the bacterial community sitting untouched on its limbs. The balance between tending and harvesting, between maintaining the crop and consuming it, is the behavioral solution that the long history of this lineage arrived at, refined over time until the behavior matched the biology of the bacteria with the same quiet precision that the waving motion matched the chemistry of the vent. The setae themselves are adapted for this relationship. Under electron microscopy, the fine structure of the setae on the chelipeds differs from that of setae found elsewhere on the animal's body. Setae on the walking legs, on the antennae, on the ventral surfaces of the body. These have the architecture of sensory or protective structures. They are stiffer or differently spaced or shaped at their tips for a particular function. The setae on the chelipeds are different, finer, more numerous, arranged in a way that provides maximum surface area relative to the volume of the cheliped itself. More surface area means more sites for bacterial attachment. More bacterial attachment means more biofilm. More biofilm means more food. The animal's body has been shaped over generations in the dark to carry as much bacterial garden as its limbs can hold. There is something very old in this arrangement. Not old in the sense of ancient lineage, though the lineage is certainly ancient, but old in the sense of being a direct, unmediated solution to a problem. The problem is energy. All living things, at every depth and every latitude and every pressure, are engaged in the continuous business of acquiring enough energy to persist. At 2,000 meters below the ocean surface, far below the zone where sunlight penetrates, far below the photosynthetically productive water where most of the ocean's food originates, acquiring energy is a more constrained project than it is at the surface. Most deep-sea animals survive on the slow rain of organic matter that sinks from above, particles of dead organisms, fecal pellets, the occasional large carcass, but this food source is sparse and irregular. At hydrothermal vents, the Earth offers an alternative, chemical energy, released continuously from the interior, available to any organism with the metabolic machinery to access it. Kiwa hirsuta does not have that metabolic machinery itself. It cannot oxidize hydrogen sulfide directly, but it can carry the bacteria that can, and feed those bacteria the gradient they need, and harvest the fixed carbon they produce. It has, in this way, borrowed the metabolism it needed. Not through genetics, not by incorporating bacterial genes into its own cells, as some organisms have done over evolutionary time, but through behavior, through the simple, repeatable act of waving its claws through the right chemistry and then scraping its mouthparts along the result. The solution is almost elegant in its directness. Find the bacteria that can do the chemistry. Put them somewhere they can grow. Keep them growing. Eat them. The waving motion observed on video from a submersible camera looks at first like something without consequence. The slow rhythmic movement of a crab's claws, back and forth, in the dim water above a dark seafloor. It does not look like agriculture. It does not look like any familiar human category of food production. There is no soil, no seed, no field. There is a crab, alone or in a loose cluster with others, holding its pale claws in the faint current above the vent, sweeping them in patient arcs through water that is chemically different from centimeter to centimeter. The motion has a cadence that submersible observers have described as deliberate, unhurried, consistent, performed at roughly the same tempo, regardless of whether the animal is actively feeding or not, as though the distinction between tending and harvesting is continuous rather than alternating, as though the crab is always doing both at once. In a sense, it is. The bacteria grow continuously. The oral appendages are capable of harvesting continuously. The waving that maintains bacterial productivity and the harvesting that removes bacterial biomass can proceed at the same time in overlapping cycles, the bacterial layer never fully stripped and never fully left to accumulate unchecked. The farm and the harvest are the same activity, performed by the same animal at the same moment in the dark water above the crack in the seafloor where the Earth's heat rises, and the chemistry that sustains everything in this community comes up from below. The bacteria that dominate the setae are not all of a single kind, though the epsilon proteobacteria, the sulfide oxidizing group that performs the primary chemosynthetic work, are the most abundant. Alongside them in smaller proportions are representatives of the gamma proteobacteria, a group that includes relatives of the symbiotic bacteria found inside Riftia tubeworms and Bathymodialis mussels. The community on the setae is a mixture, a diverse bacterial assemblage in which different organisms may use different chemical substrates or occupy different micro-niches along the length of each setae. Some positioned near the base where the thermal environment is slightly different from the tip, some tolerating higher sulfide concentrations, some requiring more oxygen. The seta itself, a few thousandths of a millimeter wide and a millimeter or two long, contains within its bacterial community a range of microbial biology that would require a laboratory full of specialists to fully characterize. The full characterization remains an open project. Molecular techniques have identified the dominant taxa, confirmed the presence of the major metabolic groups, and established that the bacterial community on Kiwa hirsuta's setae is stable enough and consistent enough across individuals to constitute a genuine biological feature of the species, rather than a random collection of environmental microbes. But the precise metabolic contributions of each bacterial taxon, the spatial organization of the community along the seta's length, the degree to which the crab actively shapes the community's composition through its behavior, these remain questions that the available methods have not yet fully answered. The picture is clear in its broad outlines and still being filled in at its edges. That incompleteness is in its way appropriate to the setting. A hydrothermal vent, 2,000 meters deep, on a ridge in the Pacific Ocean that few submersibles have visited more than a handful of times, is not a place whose biology yields easily to investigation. Each expedition that reaches this depth returns with a small increment of clarity, a bacterial sequence here, a behavioral observation there, an isotope ratio that confirms a dietary inference, and the picture of what Kiwa hirsuta actually does, how it actually lives, fills in slowly, over years of work and many separate voyages to a place that does not welcome repeated visits. What the picture shows, even in its partial state, is an animal that has closed a metabolic loop. It lives where the bacteria that could feed it are possible. It carries those bacteria on its body in a structure adapted for that purpose. It performs a behavior that maintains the bacteria's productivity. It harvests the bacteria using appendages shaped for that harvest, and it builds its own tissue from the carbon those bacteria produce, as legible in its isotope ratios as a signature at the bottom of a document. Each part of this system supports every other. Remove the waving motion, and the bacteria cannot access the chemical gradient they need. Remove the specialized setae, and the bacteria have less surface to colonize. Remove the vent, and there is no chemistry at all, no sulfide to oxidize, no basis for any of it. The crab and its bacteria and its vent exist as a single, mutually dependent system, each component necessary to the others, the whole thing assembled, piece by piece, over an evolutionary history that no one has fully traced. The image that stays, the one that the submersible cameras have returned to the surface for researchers to watch and rewatch in their ships' laboratories during long Antarctic nights or Pacific crossings, is simply the crab in the water, claws extended, waving. The motion is slow enough to be almost meditative. The water around the claws is dark, the vent below barely visible as a shimmer of thermal distortion in the submersible's lights. The crab does not appear to be doing anything. It appears to be waiting, but the bacteria on its claws are growing. Fed by the chemistry, the crab is continuously providing by moving them back and forth through the water that the earth has made different from itself. And the crab is eating continuously without urgency in the only way available to it, in this place, in this depth, in this cold and chemical dark. There are, on the floor of the Pacific Ocean above the ridge where this animal lives, creatures that carry their food sources inside their own bodies, sewn into their tissues as permanent metabolic partners. And there are creatures that filter the water column for whatever particles chance carries past. And there are creatures that wait for something larger to die nearby and sink. Each solution is its own long answer to the same old problem of energy in the dark, and each answer has lasted as long as the lineage that found it. Kiwa hirsuta's answer is to farm the surface of its own limbs, to turn the body itself into the substrate of cultivation, the claws into fields, the waving motion into the act of irrigation, to carry the farm with it wherever it goes, as long as it remains close to the chemistry that makes the farm possible. Of all the animals that have found their way to vent fields along the ridges of the deep ocean, and of all the solutions those animals have arrived at for persisting in a place without sunlight and without any but the most tenuous connection to the surface world's biology, this one, a pale furry-clawed crab waving its bacteria-covered limbs through the dark water of the mixing zone, harvesting the crop it tends with every slow sweep, may be the one that most quietly insists that life will find a way to feed itself from whatever the world around it makes available, not through drama or speed or great visible effort, through patience, through the slow repetition of a simple motion performed without knowing it, is farming without knowing it is doing anything at all except being what it is, in the only place its particular body and its particular bacteria and its particular behavior all work together without remainder. The claws move back through the sulfide-rich water, then forward through the oxygen-bearing cold, then back again. The bacteria divide, the biofilm thickens, the crab feeds, in the dark, in the deep, at the edge of the vent's warm influence, tending the only garden it has ever known, which is also itself. In the dark, where no season has ever turned, a single molecule meets another. It happens without light, without warmth in any familiar sense, without the slow green patience of a leaf turned toward the sun. A molecule of hydrogen sulfide, drawn upward through a crack in the ocean floor by the slow exhale of the planet itself, drifts into range of a protein folded into a particular shape over millions of generations of pressure and cold and chemical necessity. The protein reaches for it. Electrons move. A small amount of energy is released, not light, not heat that any surface creature would recognize as warmth, but chemical potential, the quiet currency of this place, transferred from one molecule to another in a transaction that takes no longer than a heartbeat and leaves no visible trace. And then it happens again and again. In a single bacterial cell, no wider than a few millionths of an inch, this transaction is occurring thousands of times each second in the cold dark at the bottom of 2 miles of ocean, and has been occurring without pause since long before any animal with eyes first looked at the sky. This is where the energy comes from, not from sunlight, not from the slow burning of sugars traced back to chlorophyll and photons and the ordinary miracle of a clear day. Here, at the edge of a hydrothermal vent on the Pacific Antarctic ridge, the energy that powers every living thing in the community comes from the Earth's own chemistry, from the gap between what the planet has made underground and what the ocean holds above, from the electrochemical distance between hydrogen sulfide rising and oxygen drifting down, a gap that bacteria have been crossing for longer than there have been animals to consume them. The protein that performs this transaction is called a sulfide oxidoreductase, but is embedded in the membrane of the bacterial cell, a single cell, one of uncountable billions clinging to the rocks and chimneys and sediments around every active vent field on the ocean floor. The oxidoreductase does not think, it does not choose. In the way of all enzymes, it holds its substrate and catalyzes the reaction that the chemistry of the place already wants to happen, only faster, and captures the released energy before it can simply disperse as heat. The hydrogen sulfide gives up electrons to the enzyme. The enzyme passes them along. The electrons travel a short chain and are eventually accepted by oxygen, or, where oxygen is scarce, by nitrate, which functions as a chemical stand-in for the same role. At the end of the chain, the cell has energy it can spend. It spends it on building more of itself, on fixing carbon dioxide from the surrounding water into organic molecules, on assembling the sugars and proteins and membranes that make it a living thing. Carbon dioxide enters as a gas dissolved in the cold water. It leaves as the carbon skeleton of organic life. The pathway by which some of these bacteria accomplish this fixation is called the Wood-Lungdahl pathway, and it runs as a linear chain of chemical conversions, simpler in its basic structure than the looping cycles of surface photosynthesis, older by some accounts, and well suited to the anaerobic and near anaerobic conditions that frequently occur at the boundary between vent fluid and ambient seawater. Other bacteria use the reductive TCA cycle, running the chemistry of energy generation partly in reverse to pull carbon from the water and deposit it into organic compounds. The mechanism differs between lineages. The outcome is the same. Carbon that was dissolved and inorganic becomes carbon that is bound and alive, available to be eaten, to pass upward through the food web of this small and self-contained ecosystem at the bottom of the dark. The bacterial cell divides. Its daughter cells divide. The colony expands along the surface of a seta, a single silken filament on the claw of an animal that holds perfectly still in the mixing current above the vent, moving only to bring the filament first into the sulfide-rich plume and then back into the oxygenated water above. The bacteria receive both. The enzyme fires again. The electrons move. The energy is captured. And somewhere in the slow darkness of 2 miles of ocean, without sunlight, without seasons, without any reference to the turning of the world above, the first link in a food chain is being forged one molecule at a time. The ocean floor at an active vent site is not a uniform place. Stand at the rim of what geologists call a diffuse flow zone, where warm vent fluid seeps upward, not from a single chimney, but through cracks and pores in the basaltic rock across a wide area, and what surrounds that place would appear, to an eye that could see it, as a patchwork of microenvironments so distinct that different organisms inhabit areas separated by only inches. Closest to the vent orifice, the fluid is superheated, laden with dissolved metals, and carries almost no oxygen. No macroscopic animal lives there. The zone belongs to archaea, ancient single-celled organisms, so distantly related to bacteria that they occupy their own domain of life, their biochemistry shaped by conditions that most other living things cannot survive. Some of these archaea grow at temperatures above the boiling point of water at the surface. One, known as Methanopyrus canleri, is the record holder among known life. It grows at temperatures reaching above 250 degrees Fahrenheit, at pressures that keep the water liquid despite that heat, in a chemical environment that would dissolve or denature almost any other biological molecule. Its cell wall is built from compounds not found in bacteria. Its membrane lipids are linked by chemical bonds, ether bonds rather than ester bonds, that remain stable where ordinary membranes would fall apart. Inside its cells, the enzymes that carry out metabolism are pressure stable, temperature tolerant, and configured to work fastest in conditions lethal to every creature above it in the water column. It is not adapted to extremes in the way a surface organism might gradually adjust to harder conditions. It is built for them in the way of something whose ancestors have known nothing else for longer than animals have existed on this planet. Farther from the orifice, the temperature drops steeply. Within a foot or two of the hottest zones, the temperature at the seafloor falls by more than half. A few inches farther, and it falls again. The gradient is not gradual in the way that temperature changes across the surface of the Earth, with altitude or season. It is compressed, intense, a shifting landscape of thermal microhabitats stacked within arm's reach of one another, and in each microhabitat, different organisms, bacteria adapted to warm but not scalding conditions. Then, at the margins of the vent's thermal influence, Bacteria and archaea suited to water that is barely above freezing, just a degree or two above the ambient temperature of the deep ocean floor. Each community lives at the edge of its tolerance. Each one is positioned at the exact chemical and thermal gradient that its metabolism is built to exploit. The bacteria that matter most to Kiwa hirsuta, the ones that colonize its claws and grow there in dense biofilm communities, live not at the extreme ends of this gradient, but somewhere in the middle. They are adapted to the mixing zone, the narrow band of water where vent fluid and ambient seawater meet and intermingle, where both hydrogen sulfide and oxygen are present at concentrations that support chemosynthesis, where the temperature is warm enough for their enzymes to run efficiently, but not so hot that their membranes begin to fail. This is a demanding and unstable location. The chemical concentrations shift with every pulse of vent fluid. The temperature fluctuates. The oxygen levels vary with the mixing ratio of the two water bodies. An organism living here must tolerate variability that a stable surface environment would never impose. The bacteria on the crab's setae belong predominantly to two groups. The first are epsilon proteobacteria, relatives of genera called sulfurimonas and archobacter, sulfur-oxidizing bacteria capable of using either oxygen or nitrate as their electron acceptor, which gives them flexibility in the chemically variable mixing zone where the crab positions itself. The second group are gamma proteobacteria, related to the chemosynthetic bacteria that live inside the tissues of tube worms and vent mussels elsewhere in the Pacific vent community. Those internal symbionts live in a protected and stable chemical environment supplied by their hosts. The bacteria on Kiwa hirsuta's claws live outside, exposed to the flux of the mixing zone, but supplied with chemical resources by the crab's own movement, rather than by any internal chemistry the host provides. Both strategies arrive at the same outcome: chemosynthetically fixed carbon transferred to the animal that cultivates the relationship, but the mechanics are entirely different, and the difference in exposure between an internal symbiont sheltered inside a gill cell and an external epibiont clinging to the outside of a seta in open water is the difference between a greenhouse and a field. The microbial mat that would form on a rock face near a diffuse flow zone offers a useful picture of the same biology at larger scale. Beggiato, long filamentous, white-appearing bacteria, named for a 19th century Italian naturalist, drape across the seafloor in thick mats wherever vent fluid seeps slowly through the sediment. Their white color in the dim light of a submersible's lamp comes from the elemental sulfur they store internally as a reserve of oxidizable substrate, a chemical pantry that allows them to keep metabolizing during brief periods when hydrogen sulfide is less available. They form tangible structures, not the delicate biofilms of individual setae, but visible textured mats several inches thick, their filaments interwoven and stratified in vertical layers that reflect the chemical gradient running through them. The deepest layer is richest in sulfide and poorest in oxygen. The surface layer is most oxygenated. The most productive chemistry happens in between, where the two gradients cross, and that is precisely where the densest, most metabolically active filaments are found. A current in the diffuse vent flow parts the mat surface for a moment. The filaments sway, the disturbance is slight, the thermal convection rising from the seeping warm fluid, a current no faster than the drift of a hand moved slowly through still water. In the brief parting, the interior of the mat is visible for a second. Pale layers below, the faint shimmer of heat distortion rising from a zone too warm for the surface-dwelling filaments, and then the mat closes again and returns to the steady, unwitnessed work it has been performing since long before any submersible came to observe it. What the mat does slowly across a wide area of seafloor, a single bacterial cell on the seta of Kiwa hirsuta does quickly, in miniature, in a location supplied with chemical gradient by the crab's own movement rather than by the slow geography of the vent field itself. The crab brings the bacteria to the chemistry rather than allowing the bacteria to settle where the chemistry happens to be favorable. It is an arrangement that has allowed the crab to range slightly farther from the vent than a sessile organism like a mat-forming bacterium could, to occupy positions where the chemical flux is imperfect, supplemented by the wave of the claw through air-bearing water above and sulfide-rich water below. The bacteria grow regardless. The claw moves. The gradient is maintained by motion rather than by proximity. Below the mat and the crab and the vent community, the oceanic crust is itself a microbial habitat. Water percolates downward through cracks in the basalt, circulating through the upper crust for decades before returning to the sea floor as hydrothermal fluid. In the passages through which it moves, chemosynthetic microorganisms colonize the rock walls, living at densities low enough that no visible mat forms, but present in communities that may together constitute a significant fraction of the total microbial biomass on Earth. These deep crustal microorganisms receive their chemical energy from the reaction of water with iron-bearing minerals in the basalt, a reaction called serpentinization, which produces hydrogen gas that certain archaea and bacteria use as an electron donor in the same way that vent communities use hydrogen sulfide. They live in darkness and in slowness. Their generation times may be decades. Their metabolisms run at a rate so low that they are almost impossible to distinguish from mineral processes. And yet they are alive, carrying genetic information, dividing eventually, shaping the chemistry of the fluid that will eventually reach the vent and feed the community above. The oldest biological question this place raises is not about the animals or even the bacteria at the vent surface. It is about the relationship between chemistry and life, about how far down into the planet's own processes the biological world extends, and whether the distinction between geological chemistry and biological chemistry is as sharp as the surface world suggests. At a hydrothermal vent, that boundary is genuinely uncertain. The reactions that make chemosynthesis possible, the oxidation of sulfide, the reduction of nitrate, the fixation of carbon dioxide, are reactions that also occur abiotically, without any biological catalyst, wherever the right chemicals meet under the right conditions. Life stepped into the space of these reactions at some point, brought enzymatic acceleration to them, and captured the energy they release more efficiently than the abiotic reactions could. But the reactions themselves are older than life. They were happening in hydrothermal systems on the early Earth before any cell existed, perhaps providing the chemical context in which the first self-replicating molecules found a supply of energy that did not depend on the sun. None of this is settled. The question of where life on Earth began, whether at hydrothermal vents or in warm pools at the surface, or somewhere else entirely, remains open, the evidence partial, the rocks of that era so thoroughly remade by subsequent geology that the original record has been largely lost. What can be said is that hydrothermal vents demonstrate, clearly and continuously, that life does not need sunlight, that carbon-based metabolism is possible on chemical energy alone, and that self-sustaining ecosystems can exist in complete independence from the photosynthetic world above them. Whatever came first, this strategy works. It has worked for an immense span of time. The mat on the seafloor, the epigont on the seta, the archaeon at the edge of the boiling zone, all of them are evidence of that. The carbon fixed at the vent does not stay at the vent. Some of it drifts upward in the water column as dead microbial cells, as shed fragments of biofilm, as the dissolved organic compounds that leak from living bacterial populations. Some of it is eaten by vent animals and incorporated into their tissues. And when those animals die and their bodies sink or are consumed by other animals, and those animals die, the carbon moves outward and downward, dispersed across the surrounding seafloor in a slow rain of organic particles that serves as food for the scavengers and deposit feeders of the deep ocean floor, far from the vent. The vent ecosystem is not isolated. It contributes to the organic budget of the broader deep sea in ways that are still being quantified, still being mapped, still being traced through isotopic signatures in the tissues of animals collected far from any active vent field. The tool by which scientists have traced these pathways is stable isotope analysis, the measurement of the ratio between two forms of carbon, carbon 12 and carbon 13, that are incorporated into organic molecules in slightly different proportions depending on the metabolic pathway used to fix them. Photosynthetic organisms, fixing carbon through the familiar Calvin cycle in sunlit surface waters, produce organic carbon with a characteristic ratio. Chemosynthetic organisms, fixing carbon through the Wood-Lungdahl pathway or the reductive TCA cycle in the dark, produce organic carbon with a measurably different ratio, more depleted in the heavier isotope, or in some pathways enriched in it, depending on the enzyme and the conditions. The difference between the two is small in absolute terms, but reliable enough that a single measurement of an animal's tissue can reveal whether the carbon in that tissue came from sunlight or from the Earth's own chemistry. The measurement is made in a mass spectrometer, a machine that separates molecules by their weight, counts them, and reports the ratio. The tissue sample is small. The result is a number, and that number, when plotted on a graph alongside the isotopic values measured from surface photosynthetic organisms on one side and vent chemosynthetic organisms on the other, places Kiwa hirsuta clearly and unmistakably in the chemosynthetic column. The carbon in the crab's body did not come from sunlight. It came from the oxidation of hydrogen sulfide in the dark, fixed by bacteria into organic molecules, consumed by the crab that grew them on its claws. The graph shows this without ambiguity. There is no other reading. The nitrogen isotope ratio, measured separately, provides an additional confirmation and a rough indication of the animal's trophic position, how many steps from the primary producers it feeds. The nitrogen 15 signature rises predictably with each step up a food chain because the lighter isotope is excreted slightly faster than the heavier one at each trophic transfer, leaving the animal with relatively more nitrogen 15 than the food it ate. The value in Kiwa hirsuta tissue is consistent with feeding at the first consumer level above primary production, consistent with eating bacteria directly rather than eating animals that ate bacteria. The crab is not merely living near chemosynthetic primary production. It is feeding on it at very close range, with very little trophic distance between itself and the microbes. This is confirmed by anatomy. The mouthparts of Kiwa hirsuta include specialized appendages, small, fine bristled structures positioned to draw across the seta surface and scrape the accumulated bacterial film from the filaments. Under magnification, these appendages bear rows of comb-like structures suited exactly to this task, their geometry matching the geometry of the seta they process. The crab does not consume bacterial cells one at a time. It strips them in aggregated biofilm material from the surfaces of its own claws, the biofilm that has grown slowly over the time since the claw was last cleaned. Colonies of epsilon proteobacteria and gamma proteobacteria accumulating layer by layer for fixing carbon and dividing and building organic mass from the chemistry the claw's movement continuously provides, is harvested in a single slow pass of the scraping appendage and drawn toward the mouth. The bacterial cells are then digested. Their organic carbon, the carbon they fixed from the cold water through sulfide oxidation and Calvin cycle, or Wood-Lungdahl pathway chemistry, becomes the carbon of the crab's own body, its tissues, its egg yolk, its gill membranes, its muscle. The atoms that began as carbon dioxide dissolved in seawater at 2 miles of depth, that passed through the enzyme of a sulfide oxidizing bacterium and were assembled into organic molecules by a metabolism running on the Earth's own heat, that accumulated on a silken filament in a biofilm community in the mixing zone of a hydrothermal vent. Those atoms are now the animal that farmed them, a nitrogen atom that began in the dissolved nitrate of the ambient deep ocean, used by an epsilon proteobacterium as an electron acceptor in place of oxygen during a period of low oxygen availability near the vent, incorporated into the bacterial cell's own amino acids and proteins, scraped from the claw surface by the fine bristled mouthpart of a pale crustacean at the bottom of the Pacific. That nitrogen atom now sits in the protein of a chelipid mussel, helping to move the very claw on whose surface it was first fixed. There is something in this, not a moral, not a design, but a kind of deep circularity that belongs to the way matter moves through living systems everywhere, traced most clearly here because the system is small and closed and dark and far enough from everything ordinary that its workings can be seen whole. The circularity extends outward. When Kiwa hirsuta dies, its body sinks or is consumed. The carbon and nitrogen in its tissues pass to other organisms. Some of it returns, eventually, to the dissolved pool from which the bacteria drew it. Some of it drifts beyond the vent field in particles, joining the slow descent of organic matter that rains continuously downward through every part of the deep ocean, feeding the communities of the dark water column and the soft floor below. The atoms scatter. The cycle continues. Nothing is ended by the individual death because the matter does not stop, only the particular arrangement of it that was the animal. In the vicinity of a vent, this cycling is accelerated relative to the surrounding deep seafloor, where organic input from above is sparse and slow, and the communities of animals are correspondingly thin. At the vent, primary production is happening continuously, locally, at rates that can support dense aggregations of animals, the clusters of Kiwa hirsuta individuals, the mussel beds, the shrimp congregations, the tube-worm bushes, in a small area of otherwise nearly barren seafloor. The vent is a patch of intense biological activity in the vast dark of the abyssal plain, its existence dependent entirely on the geological accident of its position above a diverging tectonic boundary, its chemistry driven by magmatic heat that rises from far below the crust. The geological processes that create a vent operate on timescales far beyond a lifetime. The Pacific Plate and the Antarctic Plate move apart by a few centimeters each year, the distance a fingernail grows in a month, sustained across 100 million years. The crust that forms where they separate is thin and relatively young, and the magma beneath it is close to the surface, close enough that seawater percolating downward through cracks in the basalt is heated quickly and driven back upward before it has traveled far. The vent fields that result are geologically transient, active for decades, or perhaps centuries before the seafloor. Spreading carries the crust away from the heat source, and the vents cool and die. New ones form elsewhere along the ridge. The community disperses or dies with the vent, depending on the dispersal capacity of its members and the distance to the next active field. Kiwa hirsuta and its relatives must find active vents to survive. The chemical gradients that the bacteria on their claws require, the hydrogen sulfide, the mixing zone, the warm but tolerable temperatures, exist only at active vent fields. The larvae that drift upward into the water column from brooding females must eventually locate a new vent or settle at the same one their mother left. How they do this is not fully resolved. The best current picture is that chemical gradients in the water column, the faint traces of hydrogen sulfide and methane that vent fluid releases into the overlying ocean as it mixes and dilutes, serve as navigational cues that draw settling larvae toward the source. The larvae follow the gradient downward, concentration increasing as they descend until they find the basalt and the warmth and the chemical richness that will allow them to begin growing their own microbial gardens. This is a journey made in darkness, at depth by an organism no larger than a grain of sand, following a chemical thread through miles of open ocean to a target it has never visited, but is, in some sense, already built for. There is no intention in it. The larva does not know where it is going. The chemical gradient simply makes the descent more likely in some directions than in others, and the descent that ends at an active vent is the descent that results in survival, and the larvae that survive are the ones that respond most strongly to the gradient. And so across generations, the response is shaped and refined and passed forward. The vent finds the larva as much as the larva finds the vent. The community that assembles at an active vent is not random. It follows a succession from the initial microbial colonizers that coat the fresh basalt with biofilm, to the small invertebrates that feed on them, to the larger crustaceans and mollusks that occupy the warmer positions. Kiwa hirsuta is not the first to arrive. It comes after the microbial mat has established itself, after the chemical gradients have stabilized enough to support the specific bacteria it cultivates. It positions itself in a zone that another smaller organism has already made hospitable, not by deliberate choice, but by the same process of chemical following that guides the larva to the vent in the first place. The community constructs its own conditions, layer by layer, the output of each stage becoming the input of the next, the way all ecosystems work, only compressed and made visible here by the small scale of the vent field and the starkness of the surrounding dark. The surrounding dark is genuinely absolute. At 2 miles of depth, no photon from the surface has ever reached the floor. The only light that exists at the vent is the faint thermal radiation of the hottest fluid, invisible to most biological visual systems, but present as a physical fact, a glow below the threshold of ordinary vision, registered perhaps by some organisms with unusual photoreceptor sensitivity, but not by Kiwa hirsuta, and whose eyes have been largely unmade by long generations in the aphotic deep. The animal lives in a world it reads not with light, but with chemistry. The fine sensors on its antennules and setae reading the concentration and composition of dissolved molecules in the water, translating the invisible landscape of chemical gradients into information about where to be, where to move, how far to extend the claws. Chemistry is this ecosystem's light. The gradient between hydrogen sulfide and oxygen is the gradient between rich and poor, between growth and starvation, between where the bacteria thrive and where they do not. The crab reads this gradient and positions itself accordingly. The bacteria read nothing. They simply grow faster where both substrates are present and grow slower where one is absent. The crab's movement, performing the function of positioning the bacteria in the gradient, does not require the crab to understand what it is doing. It requires only that the behavior that positions the bacteria well results in more energy available to the crab, and that this energetic advantage is sufficient to maintain the behavior across generations. Whatever the history of that selection, the outcome is a crustacean that farms microbes in complete darkness, 2 miles below the surface of the Pacific Ocean, by waving its claws through a chemical gradient it cannot see, cultivating life on its own body, the way the Earth itself cultivates life at every crack and seam where heat and chemistry meet the cold of the deep. The bacteria do not know they are being farmed. The crab does not know it is farming. The chemistry runs, the electrons move, the carbon is fixed and eaten and fixed again, and in the slow dark of the ocean floor, around the small, warm, chemically rich patch of seafloor where the planet's heat comes closest to the surface, a community of living things continues the work it has been doing since long before the first animal opened its first eye and looked at the sun. The vent exhales, the mat sways, the claw moves through the water in a slow arc, carrying its passengers through the gradient they require, and returns and moves again. The electrons pass from molecule to molecule in the dark. The carbon is built and rebuilt, passed from cell to tissue to body, and back to the dissolved pool from which it came. And all of it runs on the Earth's own chemistry, on the gap between hydrogen sulfide and oxygen, on the ancient reaction that no eye has ever watched and no sun has ever powered, continuing at the bottom of the cold, in the dark, in the deep, with the patience of something that has never known any other way to be. The eye of Kiwa hirsuta is a small thing, smaller than a sesame seed. Pale, faintly opalescent, set in the narrow front of the carapace, where a compound eye would be expected to sit, where, in all its relatives, in nearly every crustacean alive today, a functional eye does sit, arrayed with its hundreds of precise lenses, each one catching light and passing it inward to be read. In Kiwa hirsuta, the architecture is there, but it has been undone. What remains is a reduced structure, a few disorganized cells where the ordered array once was, the biological equivalent of a room whose shelves have been slowly cleared over generations until only the bare walls remain. Evolution does not take things away as punishment, it takes them away as economy. Where there is no light, there is no selection pressure to maintain the cellular machinery that reads light. And so that machinery, slowly, over more time than can be easily held in the mind, falls into disuse. The genes that might repair it, maintain it, build it larger, are not selected for. The genes that do other things persist. And the eye, uncalled upon in the absolute black of 2,200 meters of ocean, quietly retreats. The dark that Kiwa hirsuta inhabits is not the dark of a room with the curtains drawn. It is not the dark of a cave mouth, or a deep forest at midnight, or the inside of a closed fist. It is a darkness that has never been interrupted. The sun has never reached it. The moon has never reached it. No star has ever cast even the faintest, most attenuated photon to that depth without having it absorbed long before arrival. The darkness at the bottom of the Pacific Antarctic ridge, where the crust has opened and the heat of the interior comes through, is the oldest kind of dark, the dark that existed before there were eyes to notice it, the dark that will persist long after. And into that dark, Kiwa hirsuta moves with a precision that has nothing to do with sight. The antennules of the crab are modified structures, finely tuned for reading chemistry in the water. They are not searching blindly. They are reading gradients, the slow increase of hydrogen sulfide in one direction, the trace warmth of diffuse vent flow rising from a particular seam in the rock, the faint chemical signature of other members of its own kind, gathered at the margin of an emission. The mechanoreceptive hairs distributed across the body read the pressure of water movement, the slight changes in current that signal proximity to a thermal plume. The whole surface of the animal is a sensory field. It does not need to see the vent to find it. It feels the vent, chemically and physically, across a distance, and it moves toward what it senses with a directness that would seem purposeful to any observer. But nothing in this movement is purposeful in the way a mind would be purposeful. There is no intention here, no decision. What moves Kiwa hirsuta toward the vent is older than intention, a set of sensory thresholds and motor responses written in the animal's nervous tissue over immeasurable time, each one tested and adjusted by the plain fact of survival, so that the animals whose chemistry reading was slightly sharper, whose movement toward the gradient was slightly more precise, tended to reach the chemical resource a little sooner, tended to eat a little more, tended to reproduce a little more often. What remains here, now, is the accumulated result of that long process. A crab that finds what it needs in the dark without being able to see it. The callipeds extend as the animal nears the emission margin, both claws fringed along their upper surfaces with the dense, soft, yellowish setae, the bristled filaments that give the species its common name, that give the whole genus its common name, lift slightly from the body and begin their movement. The wave motion is slow, deliberate, in the way that water is deliberate, not because anything has decided, but because the rate is set by the current and the gradient and the threshold, and those things have their own pace. The claws move outward, extending into the warmer, more sulfide-rich upwelling from the vent below, then draw back through the cooler, oxygen-richer water from above, out and back, out and back. The seta catch both. They are positioned between two chemical worlds, one rich in hydrogen sulfide, one rich in oxygen, and on their surface, in the biofilm that coats every filament from base to tip, the bacteria that live there receive exactly what they need. Sulfide arrives in one phase of the motion, oxygen arrives in the other. The bacterial cells fix carbon, divide, grow, accumulate along the bristle surfaces in communities as dense as any managed agriculture. The claw does the work of cultivation without knowing that it cultivates. The setae are a farm, tended by motion, harvested by the fine comb of the oral appendages, and none of it requires awareness of any kind. There is something in this that asks for a long pause, not because it is improbable, it is in its own deep way entirely logical, but because the distances between what it resembles and what it is are so great. It resembles farming in the way that a crystal resembles a building. The structure is there, the repetition is there, the output is there, but the mechanism is entirely different, and the difference runs all the way down to what life is and what a mind is, and how far apart those two things can be, while still producing results that look from a distance like choice. The bacteria on the setae of Kiwa hirsuta belong largely to the epsilon proteobacteria, a lineage of microorganisms found at vent sites around the world, adapted to the sulfide-rich, oxygen-marginal conditions of diffuse vent flow. They oxidize hydrogen sulfide. They fix carbon from carbon dioxide. They build their own organic matter from inorganic sources, the way plants build theirs from light and air, but slower and colder, and in a chemistry that would poison a plant instantly. The bacterial cells are individually invisible, a few micrometers long, packed along the seta surfaces in the intimate contact of a biofilm, their outer membranes touching, their metabolic products diffusing into the thin water films between them. Each cell is doing a version of the same thing. If hydrogen sulfide arrives, an enzyme catches it, a sulfide oxidoreductase, a protein structure folded into exactly the right shape to hold the sulfide molecule against an electron acceptor and strip electrons from it. Those electrons move through a chain of protein complexes embedded in the cell membrane. At each step, their movement drives the pumping of ions across the membrane, building a gradient of charge that the cell uses to make ATP, the chemical form of energy that cells spend to do everything they do. And at the end of that chain, the carbon is fixed. Carbon dioxide from the water becomes organic carbon. The cell grows. One cell, then two, then four. The colony thickens on the seta surface. The biofilm deepens, and the claw continues its slow arc through the water, bringing the colony what it needs, keeping it alive in the place where the two chemical worlds meet. At a pressure of 220 atmospheres, a measure of weight that has no ready equivalent in any ordinary experience, except to say that every square inch of the crab's outer surface bears a force that would crush any air-filled space instantly. The tissues of Kiwa hirsuta function as though the pressure were entirely normal, which, for this animal, it is. The cell membranes are composed of a particular balance of lipids, fats, and fatty acids in a ratio that keeps the membrane fluid at high pressure, the way one might imagine adjusting the consistency of a material to keep it workable in extreme cold. More unsaturated fatty acid chains, curved and kinked rather than straight, preventing the membrane from packing too tightly, preserving the fluid state that allows the proteins embedded in it to move and function. The enzymes that carry out metabolism have their own adaptations, shapes that remain stable under pressure, that would cause a surface organism's equivalent proteins to change their conformation and lose their activity. The pressure that would kill is simply the medium the animal lives in. It has always been the medium. It is not experienced as a threat. It is as unremarkable to Kiwa hirsuta as the weight of the atmosphere is to any creature that has never known anything else. Through the gill chamber, water moves with each ventilation cycle. The gills are pale and finely structured, feathery filaments through which blood flows in close contact with the surrounding water, exchanging oxygen inward and carbon dioxide outward. But the water that flows through the gill chamber near the vent margin also carries hydrogen sulfide, and hydrogen sulfide at sufficient concentration interrupts the chemistry of respiration in the mitochondria, the small structures inside each cell, where ATP is made using oxygen. Something in the gill tissue of Kiwa hirsuta processes this incoming sulfide before it can reach the concentration that would cause harm. The most likely mechanism involves enzymatic oxidation, the same basic chemistry the epibiotic bacteria perform on the setae surfaces, but now occurring in the gill tissue itself, converting sulfide to less damaging forms before it penetrates deeper into the body. The gill chamber may also, in a secondary way, harbor bacterial colonizers of its own, not the dense farming community of the chelippids, but a surface epibiotic community nonetheless, using the sulfide that passes through as their own metabolic substrate, perhaps contributing slightly to the detoxification that protects the animal's respiratory tissue. This is not settled. The gill chamber communities have been examined, but not fully resolved, and the picture that exists is preliminary. What is clear is that the animal manages the sulfide, that it lives routinely in a chemical environment that would not be tolerable without some form of physiological defense, and that this defense is built into its biology as completely as the modified membrane, lipids, and the vestigial eye. Somewhere beyond the margin of the vent field, in the cooler, darker, less chemically complex water that surrounds the site of heat and emission, females have been observed carrying eggs. The abdomen curls inward and downward, holding the clutch of pale, smooth eggs against the ventral surface of the body, protecting them in the space between the pleopods and the underside of the carapace. The females found in this brooding condition are not, for the most part, found at the active vent margins where the adult animals spend their feeding time. They have moved outward into water that is cooler and less sulfitic. Why eggs would benefit from removal from the vent environment is not difficult to imagine. An egg is a small enclosed system, a volume of fluid inside a membrane holding a developing embryo with no capacity yet for the physiological defenses the adult has spent its whole life refining. The hydrogen sulfide that the adult detoxifies through gill tissue and enzymatic processing might be simply toxic to an embryo whose defenses have not yet formed. The temperatures immediately around active vent emissions, high enough to create a mixing zone where the water shimmers with thermal distortion, might exceed what the early embryo can tolerate. The cooler, quieter water outside the active field offers conditions less hospitable to the adult's feeding, but safer for what the adult carries. What the larvae do after hatching is not known. How they find their way back to a vent field, whether they use chemical gradients, temperature cues, pressure differences, or some combination of signals, is not known. The larval stage of Kiwa hirsuta has not been directly observed. What exists is inference. The larvae must travel, at some point in their early life, from wherever they hatch into the open water above or around the vent field, and eventually settle somewhere with the chemical and thermal signature of diffuse vent flow. How they locate that target across a distance is one of the questions the animal has not yet answered in any language that the researchers who study it can fully read. The patience required to study an animal at 2,200 meters is not the ordinary patience of fieldwork. It is the patience of repeated, expensive, technically complex expeditions, each one dependent on a vessel, a submersible, a team, a window of weather, and the continued existence of the vent field itself, which is a geological feature and not a permanent one. Vents open and close on timescales of years to decades. A vent site visited one year and revisited years later may have changed in ways that rearrange or displace the community that depended on it. The crab that was there may have followed the chemistry to another emission nearby, or may be gone entirely from that patch of seafloor. The submersible descends in the dark. Its exterior lights cut a narrow cone through the water, a few meters of pale illumination against the surrounding black. The pilot and observers watch through a small viewport or through a camera feed as the seafloor rises slowly into view. Dark basalt, pale bacterial matte, the shimmer of diffuse flow above a seam in the rock. And then, if the vent is active, the animals gathered at the margin in numbers that vary with the richness of the emission, some spreading loosely across the surrounding substrate, others pressing close to the warmth and the chemistry that the setae on their claws require. Several individuals, in the light of the submersible, hold their calipids extended and raised. The claws move, the slow arcing motion that looks almost ceremonial in the dimly lit footage. 3 or 4 animals positioned at slightly different distances from the same emission, each working its own position in the gradient, each with its own slight variation in the arc of the wave, the amplitude, a function of where in the gradient that individual sits. The setae catch the current. The bacteria on the setae receive what they need. The animals wave their claws in the dark, and the dark continues around them, and the vent continues below them, and the water continues above, and the whole system, crab, bacterium, chemistry, geology, runs on without requiring any light at all. This is what adaptation over deep time makes, not drama, not effort, not even awareness. It makes a fit, a precise, intricate, completely unintentional fit between an organism and the particular conditions of the place it occupies, so thorough that the organism is no longer separate from those conditions, but is instead a functioning part of them. Kiwa hirsuta belongs to the vent field, the way the vent field belongs to the ridge, not by design, not by preference, by the accumulated consequence of every generation that lived long enough to reproduce in this particular darkness, in this particular chemistry, at this particular depth, under this particular pressure, with these particular claws, and these particular bristles, and these particular bacteria, all of them, the animal, the microbe, the chemical gradient, the geological feature, parts of a single, slow, ongoing system that the word ecosystem only approximates. There are other members of the genus Kiwa, now animals collected from Antarctic vents, from vents along the Costa Rica margin, each one exhibiting its own variation on the themes the family carries, the setose appendages, the chemosynthetic epidents, the life organized around diffuse vent flow. Each new species found at a new vent site is evidence that the cavadi are not a local peculiarity, but a lineage that has spread along the system of mid-ocean ridges, finding vent fields and colonizing them. The family's history written in the geography of the seafloor itself. But those animals are their own stories. Cavadi hirsuta, the first described, the one with the pale cream carapace and the yellowish-white furled bristles, and the vestigial eye looking at nothing, remains the point of origin, the animal that first made the family name necessary. The family Kiwidai is a rare category. Most animals discovered today, even ones that are new to science, fit somewhere into the existing arrangement. A new species of barnacle, a new genus of snapping shrimp, a new variant of something already broadly known. These are found regularly, and they find their place in the existing framework without requiring the framework to expand to accommodate them. But Kiwidai hirsuta had no framework. The morphology of its chelipeds, its cetanution, the arrangement of its mouth parts, the form of its carapace, the structure of its gills, none of these fit any known family closely enough for placement to be reasonable. The organism stood outside all available categories, and so a new one was made. A new family in the crustaceans is an event, not a dramatic one. It happens in the pages of a scientific journal, in precise technical language, with measurements and illustrations and careful comparative statements about why the existing families are insufficient. But it represents a genuine shift in the map of life, as it is understood. The recognition that a lineage exists, whose distance from all other lineages exceeds what any existing category can hold. Kawaii now stands in the formal arrangement of living things as its own distinct and valid branch, as real as any of the thousands of crustacean families described before it. The ocean floor, far from the vent field and its gathered communities, is cold and very nearly still. The ambient temperature in the deep water of the Pacific and Arctic region is close to the freezing point of seawater, slightly below the freezing point of fresh water, kept liquid by the pressure of the ocean column above. Almost nothing lives there, by comparison to the vent margins, and the organic matter that settles from the surface, the slow rain of dead cells, shed scales, the remnants of organisms that lived and died in the upper water column thousands of meters above, falls across the deep sea floor in a thin, continuous, almost imperceptible drift that the organisms of the abyss live on, very slowly in a metabolism calibrated to near nothing. But at the vent margins, The floor is alive in a way that the surrounding seafloor is not. The biomass concentrated at a single active vent field is out of proportion to its surface area, a dense, warm, chemically complex island of productivity in an otherwise sparse expanse of cold, dark ocean. And at the center of that island, or rather at its edges, where the chemistry of the vent meets the chemistry of the open sea, the crabs hold their bristled claws in the current and tend the living film that coats them. The body of the crab itself, 15 centimeters from the front of the carapace to the end of the abdomen, roughly the length of a large hand, is unremarkable in its broad outlines. A crustacean body, jointed legs, a carapace that covers the vital organs, an abdomen that flexes, gills inside a chamber, a heart, a gut, the basic plan that the Decapoda have carried through hundreds of millions of years of ocean, refined and adjusted in thousands of different directions by thousands of different sets of conditions, producing the shrimp and the crab, and the lobster and the hermit crab, all of them variations on the same basic architecture, the same fundamental relationship between the exterior world and the chemistry inside. In Kiwa hirsuta, that architecture has been shaped by the particular demands of the vent environment into something that looks familiar. A crab is recognizable as a crab, but functions in ways that no surface crab does. The eye retreated, the sati elaborated, the gill tissue developed its sulfide tolerance, the membrane lipids adjusted for pressure, the oral appendages became fine-combed for scraping. Each change was small, measured against the whole. Each change was the residue of a long process of differential survival. Together, they make an organism that is precisely here, precisely at this depth, in this chemistry, on this geological feature, and nowhere else. The claws wave in the dark. The bacteria grow. The vent breathes upward through the crack in the crust, carrying the chemistry of the Earth's interior into the column of cold ocean water above, and the water carries it outward, and the gradient across which the claws move, sulfide below, oxygen above, is replenished continuously as long as the vent is active. The crab does not know this. The crab does not know anything, but the crab is alive in this gradient, sustained by it, shaped to it, over time beyond easy counting, and the bacteria on its claws are alive in the same gradient, shaped to it, in their own way, each organism's history braided with the others in a relationship older than either species in its current form. The relationship between Kiwa hirsuta and its epibiotic bacteria is not symbiosis in the sense of mutual benefit between two individuals who have entered into an arrangement. No arrangement was made, no benefit was negotiated. What exists is the result of two sets of conditions, the conditions that favor bacteria that can colonize a moving, sulfide-exposed surface in warm vent-adjacent water, and the conditions that favor a crab that can harvest those bacteria and digest them, aligning over long time into something that looks, from the outside, like a partnership. It is a partnership in the same way that a river and a riverbed are partners. Neither chose the other. Neither benefits in a way that required choice, but each is shaped by the other, and what they make together is something neither makes alone. This is perhaps the deepest quality of life at the vents, not that it is extreme, not that it is improbable, but that it is ordinary, in the way that all life is ordinary from the inside. Kiwa hirsuta is not straining against its environment. It is not surviving despite the darkness, the pressure, the heat, the chemistry. It lives within all of these the way a reef fish lives within the shallow, warm water of a tropical coast, as the native condition, the expected state, the thing that feels like nothing because it has always felt like exactly this. Outside the vent, The seafloor extends in all directions into the dark. The ridge continues for thousands of kilometers, its elevated spine rising above the surrounding abyssal plain. The cracked and recracked again basalt cooling slowly as it moves away from the zone of spreading, crystallizing fully as it goes, carrying whatever lives on it outward at the pace of centimeters per year. Other vent fields mark the ridge, some known, some not yet found, each one a small bright island of chemistry in the cold dark, each one potentially hosting its own community of organisms adapted to the particular expression of heat and fluid chemistry that issues from that particular crack in that particular section of the floor. On some of those vents, Kiwa hirsuta or its relatives may already be present, living out the same rhythm that the animals at the Easter Island microplate vent field live. The wave of the claws, the growth of the bacteria, the slow harvest, the gathering of multiple individuals at the emission margin, each one positioned in its own pocket of the gradient. On others, the community may be different in composition, the thermal and chemical profiles supporting different organisms, different primary producers, different animals in the secondary trophic position that Kiwa hirsuta fills at its own site. The vents are not all the same. The communities they support reflect their differences, but the principle is the same across all of them. Chemosynthesis as the base, the Earth's own chemistry as the energy source, life organized entirely, without reference to sunlight. The sunlight, 2 kilometers and more above, reaches the surface of the ocean in the usual way. It warms the surface water. It drives the phytoplankton in the upper few hundred meters to fix carbon, to divide, to feed the food webs of the illuminated ocean. The zooplankton rise and fall through the water column in their daily migrations. The fish follow, the whales follow the fish. The whole sunlit economy of the upper ocean churns on in its productive, photosynthetically grounded way, filling the water with life and death and the organic matter that slowly settles downward toward the floor. But at the floor, where the vents are, none of that matters to Kiwa hirsuta. The settling organic particles from above are too sparse, too dilute, too nutritionally marginal to sustain an animal of that size at that depth. What sustains it is the vent itself, the chemistry of the Earth's interior, converted into bacterial biomass by the bacteria on its claws, converted into animal tissue by the crab's digestion, cycling the elements of life through a system entirely of the deep ocean's own making. The stable isotope ratios in the tissues of Kiwa hirsuta tell this story in numbers. Carbon fixed by chemosynthesis has a measurably different ratio of carbon-13 to carbon-12 than carbon fixed by photosynthesis. When the tissue of an animal is analyzed for this ratio and the value falls in the chemosynthetic range, it is telling the history of where that carbon came from, which chemistry made it, which organisms processed it, which chain of consumption brought it to the tissue that now holds it. The tissues of Kiwa hirsuta fall in the chemosynthetic range. The carbon in the crab's body came from the vent. It came from the bacteria. It came ultimately from carbon dioxide dissolved in deep cold water, fixed by bacterial enzymes in the dark, accumulated in biofilm on bristling claws, scraped loose by fine comb-like mouthparts, swallowed, digested, built into muscle and membrane and reproductive tissue. The vent is the crab. In the most direct chemical sense, what Kiwa hirsuta is made of was made at the vent by organisms living on the crab's own surface, processed by the crab's own body, and now held in the form of a living animal moving through the gradient at the edge of the heat. It is a closed loop, a small one by the standards of ecosystems, but complete. The claws continue their arc out through the warmer water below. back through the cooler water above. The motion is slow enough that a casual observer might not notice it at first, might see only a pale crab standing at the margin of a diffuse vent flow, its claws slightly raised, its body tilted gently toward the current. Only with sustained watching does the rhythm become apparent. Only then does the waving resolve from stillness into pattern, from a posture into a behavior, from a physical state into a process, with a beginning and a continuing and an effect. The darkness around the animal is absolute, and it has always been absolute, and it will remain absolute long after the animal that moves through it now is gone. The vent will close. Eventually, the geological forces that opened it will shift, the heat will diminish, the flow will slow and stop, and the community that gathered at this particular crack in this particular ridge will disperse or decline. Other vents will open elsewhere along the ridge, as they have been opening and closing for as long as the ridge has existed, which is longer than there have been crustaceans in the ocean to colonize them. Kiwa hirsuta will find what it has always found. The chemistry will be there. The gradient will be there. The bacteria will colonize whatever surface offers itself in the right temperature, the right sulfide concentration, the right exposure to the two chemical worlds the seedi need. The claws will wave. The farm will grow. The oral appendages will harvest, and the animal will live on what its own body has cultivated, in the dark, under the pressure of an ocean it has never seen the surface of, in water that has not been touched by sunlight, in the time it took to travel from the surface to this depth. The form of the crab is quiet. The movement of the claws is slow. The water around the vent margin moves in its steady thermal drift, warm below, cool above, and the animals positioned in it wave their bristled claws through the invisible boundary between the two, and the bacteria on the seedi receive what they need, and the system continues, as it has continued, as it will continue, patient and precise, and entirely unaware of itself, the deep ocean's small, furry, pale-eyed farmer, tending a living harvest no eye will ever see, in a dark that has no end. The cold comes first, above the vent margin, where the ambient water settles back to its natural temperature. Then the stillness, the thermal shimmer fading, the faint distortion of the mixing zone no longer visible. The basalt of the seafloor extends away from the emission in all directions, cooling by degrees, covered in the thin film of bacterial mat at the vent's edge, and bare beyond it, dark stone under dark water, under dark water, under more dark water, all the way to the surface that Kiwa hirsuta has never needed. Somewhere in the bristles of the claw, in the thin water film between two bacterial cells, a molecule of hydrogen sulfide arrives and is caught, an electron is passed, a carbon atom is fixed,